planning and design, or the proposal lab, and the implementation of that proposal, planning and design, happens to be a problem for many students and in some cases, teachers. In this video, I'm going to be taking you through the proposal and implementation as would have been written by CXC and should provide guidance to those persons who are struggling with this part of the SBA. You want to stay with me as I go through this entire lab. And of course, please be reminded to like, share, and subscribe to the channel. You can find us on YouTube at CSEC Biology TCP, or you could visit our website at tcp-academy.teachable.com. We're going to be looking at the planning and designing first, and we're going to be looking at exemplar 1. And of course, you'll see part A for the proposal or for the planning and design, and part B for the implementation. It's pretty simple. So having looked at this, you should be able to write up your planning and design. You should be able to write up your implementation with great ease. Please be reminded that the implementation must be submitted as one of the AI lab to be submitted. Let's hop right into the lab. We're looking at the planning and design. So it begins with observation. Now, 10-year-old John observed that after his grandfather planted some bean seedlings, he immediately applied a blue liquid to them, which he had carefully measured out into the watering can. He asked his older siblings what was the blue liquid their grandfather applied to the seedling and why did he measure it. All right, so I am assuming that the blue liquid here is copper sulfate. It's a type of fertilizer. So the next thing to do here to find out is to formulate an hypothesis. The hypothesis here is get help with CSEC Biology SBA Labs and Human and Social Biology SBA Labs at tcp-academy.com. Tim had questions. He wanted to know where he could get help with his SBA labs. His parents searched but could not find that experience. Tim also made some calls, however could not find the help he needed. Tim needed an experience he could trust to help him get over the hurdle of SBAs as success was in sight. He met the guys from tcp-academy.teachable.com and they designed a program to help him. tcp-academy.teachable.com was able to help him. So too, they can help you. Sign up tcp-academy.teachable.com for help with your SBAs in biology and human and social biology. Increasing the concentration of fertilizer applied to bean seedling increases the number of leaves produced in the bean seedlings. The next step is to formulate an aim, and the aim must have a direct relation to the hypothesis. Here's the aim: to determine if increasing the concentration of artificial fertilizer increases the number of leaves produced in the beet seedlings. After we would have established that, the next step is to write out your material/apparatus. Now we're going to need clean wash sand, distilled water, 5 beakers, red beans, 5 plastic trays of the same dimensions, foil trays, 4 measuring cylinders, a liquid fertilizer. So this is all clearly stated with even a number of each that you'll need. as somebody else might be the person responsible for carrying out the lab. Let's look at the method and how the method / procedure is somewhat different than it would have been for the rest of labs you have been writing. All apparatus will be cleaned and dried before beginning the experiment. Here you observe that we are writing in future tense. What is it that we are going to do? What is it that is expected to be done? The 4 trays will be labeled as follows. No fertilizer, that's one of the tray. That seemingly is your control. 3/4 strength, half strength, quarter strength. Here you realize that there is a clear demarcation as to how the condition to which the seeds are going to be exposed, and it is well outlined there in your procedure. Then it says, take the fertilizer and make it up to full strength, following the manufacturer's instructions. Make up to 1 liter. Label this full strength. In everything you do, you are going to be labeling to ensure that you minimize the errors that might be associated with your experiment. Make up dilute solution of a fertilizer as follows. Look at this. Measure out 150 ml of a full strength into a beaker. Using a measuring cylinder, measure 50 ml of distilled water and add to the beaker. Label this 3/4 strength. Look at what is happening. Measure out 100 ml of a full strength into a beaker. This is a separate concentration. Using a measuring cylinder, measure 100 ml of distilled water and add to the beaker. Label this half strength. Measure out 50 ml of full strength into a beaker. Using a measuring cylinder, measure 150 ml of distilled water and add to the beaker. Label this quarter strength. Fill the tray with washed dried sand. In each tray, plant 4 beans. I hope you are following the detailed instruction. Each bean should be planted no more than 1 centimeter below the surface and should be spaced as far away from each other as the container will allow. Then you are going to saturate the soil in the tray labeled no fertilizer. How are you going to do this? By adding measured amount of distilled water until the sand is moist. Add the same volume of distilled water to each of the other trays. So you want to ensure that here everything is starting out pretty much with the same amount of water. You would appreciate the fact that water is needed for hydrolysis in germination. Now, to tray labeled no fertilizer, you are supposed to add 15 ml of distilled water. Now the experiment really starts. To the tray labeled full strength, measure out 15 ml of the fertilizer labeled full strength. That's a solution labeled full strength. Repeat the procedure for the remaining trays. Repeat the addition of a 15 ml of liquid to the appropriately labeled tray for the next 10 days. So here it is telling us that this is not a 1-day thing. It is going to take us 10 to 11 days to complete this experiment. Ensure that the solution is added at the same time each day. We want to ensure that the results of this experiment is not by chance. So we want to increase the accuracy by ensuring that everything is done at the same day, at the same time for all the sets in your experiment. Place trays in a bright, well ventilated area. Observe the tray each day. Record the day on which the beans germinated. Count the number of leaves on each seedling and record in a table. A table is always nice to use in recording your observation as it provides a quick summary, or you can get a summary at a glance looking at a table. Observation such as the color of a leaf and stem and the size of the leaves can also be recorded. Now, these are very, very important as these will help you to determine whether or not you are seeing growth. Expected result. What is it that we're expecting from all this setup? It is expected that the tray containing full strength fertilizer would have the greatest number of leaves, followed by the 3/4 strength, then the half strength, then the quarter strength. The tray containing no fertilizer should have the least number of leaves. And here it is fair to say that the one without fertilizer is our control because it has not been altered, and it will give you a true reflection as to what would have happened had the other trays not been altered with the different strength of fertilizer. So that pretty much completes the planning and design or the proposal. Now we are going to implement it. So now we're going to be looking at part B, the implementation. Here we go. Plants take up water and mineral salts from the soil. This is your introduction or the background knowledge. The mineral salts are required to ensure proper growth of plants. Nitrates, phosphates, potassium, iron, calcium, and sulfate are some of the minerals required, and they can be found in artificial fertilizers but must be applied in the amount required by the plant. The number of leaves produced by seedlings in a given time changes in length, mass, and surface area can be used to demonstrate growth in plants. In this experiment, the relationship between the quantity of fertilizer added and the growth rate of a seedling will be explored. And that nicely gave the introduction to your experiment in 246 lines. How easy that was. We're now going to be writing our procedure or method. And remember, this is just pretty much a change in tense. It's the same method that you had for the planning and designing or for the proposal that you're going to be using in the implementation. Look at the change in tense. All apparatus was cleaned and dried before beginning the experiment. The 4 trays were labeled, and you're seeing them 1, 2, 3, 4. The fertilizer was collected and made up to full strength following the manufacturer's instruction. 500 ml of solution was made up. This was labeled full strength. And you're observing here, we are following the procedure that we had in the planning and design. So you can see there 1, 2, 3. Just look at it carefully. You can pause your screen at any time to read what is there and compare it to what you have in your experiment. So it's clearly stated here in 4 what is expected of you in the procedure. And remember, in the interest of time, you can pause the screen to, of course, write off the procedure or to acquaint yourself with the procedure. The expected result here in the implementation, we don't have expected result. Instead, we have result. So what is it that we observe from the experiment that was carried out? I want you to observe here that your table has a title, header rows, and header column, all well stated. I want to point out as well that the units are not repeated in the cells. So total number after 10 days. All right, so after 10 days, we had 18, 45, 33, 27, 22, so on and so forth. So you're seeing here, based on the expected result, it seems as if it is coming off to be true. Please take the time out to read, pause, and you can read at your leisure. After the result, we have discussion. And now we're going to be answering the which, where, why, when in the discussion. So it starts by saying plants need the minerals to provide the elements needed to make constituents such as protein, DNA, chlorophyll, and cellulose. Magnesium is an important part of a chlorophyll molecule required by the plant to photosynthesize. Now, if you observe the discussion here, it is not just something that is grabbed from the brain. It is something that is well researched. The information here does not have any fluff. All that it has is information that is driven by data. So here you're realizing that the information is coming from your nutrition, the topic nutrition. You're seeing something about enzyme. You're seeing the various minerals needed by the plant and what they're used for. So it's very important that you have a good grasp of the link that there is with the lab that you're doing. Where am I going to get the information to support the discussion? In some cases, it might be one topic. In other cases, it might be more than one topic. So you have to make sure that you have that all sorted out from the time you would have written your planning and design. You want to start the reading around that to ensure that you would have written an awesome discussion. After the discussion, you are going to be having your conclusion. And like I said, the conclusion is your final position. Increasing the concentration of fertilizer applied to bean seedling increases the number of leaves produced in the bean seedlings. And I want you to pay attention here to the limitations. Remember, we said that the limitation refer to those things that pretty much you have no control over. However, they could have altered the result of experiment. Please be reminded that you can pause the video to read and make that comparison with what you would have written for your lab. After your limitation, you are going to be having the reflection. And the reflection pretty much tells about what you would have learned, what new you would have picked up. I want you to look at the second paragraph here. This practical is based on section B, life processes and disease, nutrition, specific objective 2.5, and specific objective 8.1. So you are seeing here, it is telling based on the reflection, we can say exactly where in the syllabus this lab was taken from and what objective or objectives were addressed in this lab. So here we're looking at nutrition and we are looking at disease. Of course, your lab says nutrition and disease. Specific objective 2.5 and growth, specific objective 8.1. So growth came from specific objective 8.1 and the nutrition part of a lab came from specific objective 2.5, all must be documented in your reflection. Then I want you to look here at what is happening, and I strongly suggest that you make for yourself a mirror of this lab, looking at the things that were highlighted in this lab so as to ensure that you have something similar for your lab as you get better and to grow with implementation and planning and design. So be reminded at any time you can pause this video and of course look at the content that was presented. Remember that it's not as easy as you think, but it is not something that you're going to get done overnight. You need to put some reading into your lab. Remember, the lab is around disease and nutrition, and you need to have a good understanding as to where in the syllabus the lab is linked to. You need to have that understanding. So we go right back to our top of this presentation. And while I do so, I remind you to like, share, and leave a comment on the video. I do hope that you are going to be scoring all A's in your labs. I do hope that you are preparing for the examination. And I do hope that all your dreams will come through. Be reminded, you can join us at our website at tcp-academy.teachable.com. I am Mr. Wilson, and until we next meet, make sure that you would have visited the website or visited my YouTube channel to look at least some past paper. Take care. Get help with CSEC Biology SBA Labs and Human and Social Biology SBA Labs at tcp-academy.com. Tim had questions. He wanted to know where he could get help with his SBA Labs. His parents searched but could not find that experience. Tim also made some calls, however could not find the help he needed. Tim needed an experience he could trust to help him get over the hurdle of SBAs as success was in sight. He met the guys from tcp-academy.teachable.com and they designed a program to help him. tcp-academy.teachable.com was able to help him. So too, they can help you. Sign up tcp-academy.teachable.com for help with your SBAs in biology and human and social biology.